Welcome to Brilliant Katha. Today we are going to discuss most important and previous year's questions from the biology chapter Heredity and Evolution. So let us discuss, those important questions. What is the first question? First question we are going to discuss here, that is, what is gene and mention its role. What is the function of gene? Okay, so let us discuss what is gene. Here we have discussed, in human body, not only human body, all organisms, their body is made up of many cells, which is the structural and functional unit of an organism. We have discussed, right? Cell. So now, when you talk about the cell, cell has a nucleus. Now, inside the nucleus, you can see a small ribbon-like structures. Can you see the small ribbon-like structures? This is called as chromosome. Those chromosomes are seen inside the nucleus. Now, what is there inside the chromosome? On the chromosome, you can see there are small, thread-like structures. This is what? Those thread-like structures, we call it as a DNA. Now, still, we are going to discuss what is gene, not the DNA. Now, gene, where is gene? Genes are the particular part of a functional segment of a DNA. A particular part of DNA is called as gene. Now, what is the role? That is what we are going to discuss now here. Look at here. In this case, you can see here, chromosome as consists of long thread-like DNA. We have already discussed this chromosome. They have a thread-like DNA, right? Okay. Now, in this case, look at here. It contains many genes. When you talk about the DNA, it has many genes. Look at here. This is that. Okay. It can be many genes. Now, here, these genes are the functional segment of DNA. Now, why it is called functional segment or functional part of DNA? Because it has a particular role to play. What is the role? It is responsible for a particular trait. Now, this DNA what we are discussing, this gene what we are discussing, they are going to produce, this gene what they are going to produce, they are going to produce protein. Now, what is the function? What the protein will do? Protein will control a particular trait. Trait means character. Is there any character which is passing from parents to offspring? We call it as trait. So, simply means the gene which is responsible for a particular trait or a particular character. Genes are playing an important role. That is what we have discussed here. What is gene? It is the functional segment of DNA. And what is its role? It controls or it is responsible for a particular trait. Okay, that is the first question. Now, let us discuss the second question. Yes. What is the difference between autosome and sex chromosome? Now, what is autosome? Where we have discussed, autosomes are the chromosome which determine the somatic characters of an organism. What is somatic mean? Somatic mean a body character or body. Somatic in the sense body. Now, you must have seen, the human chromosome we have discussed, we have 23 pair of chromosome, right? 23 pair of chromosome. Out of that, these are the 23 pair of chromosome, which you can see, human chromosome. Now, here, in this, out of the 23 pair of chromosome, first 22 pairs are called as autosome. Now, why it is called as autosome? Because they will be having this 22, look at here. First 1 to 22 pair of chromosome, they are controlling our somatic character or body character. Like maybe they will be controlling the height of the person, maybe controlling some, shape of hairstyle or the shape of the nose, or even sometime, ear lobe is attached, sometime it is free. All those body characters are controlled by these autosomes, we can say. Autosomes are controlling. That means body chromosome, or we call it as body chromosome. Now, then, what is mean by sex chromosome? Because one of the question is that, right? What is the difference between autosome and sex chromosome? Now, sex chromosomes are which determine the chromosome which determine the sex of the individual. Because whether the person is male or female, who determine? That is the last set. Last set. We call it as a 23rd pair. 23rd pair of the chromosome, it is considered as a sex chromosome. Now, you can see the 23rd pair. This is that. If it is a, like male, the chromosome will be XY, like this. Right. If the person is female, the chromosome will be XX. Both the chromosome will be XX. So that is the main difference between autosome, simply means which determine the somatic character or body character, and the sex chromosome which determine the sex of the individual, Okay, let us go to the next question. Yes, next question is that a red flower plant is denoted capital R. Okay, it is crossed with a white flower color which is denoted by small r. So that means what we are going to do here, look at here, in this we are going to do a red flower plant. We are going to cross with a white colored flower plant. Okay, now here, in this case, look at here. From this, what is the gene? Here we are clearly said the red flower mean their gene is denoted as a capital R, and the white one is small r. Okay, now what is going to happen? Now look at here. When we cross it from here, you can see from here capital R comes. From here small r comes. Because why it is? Because they have both the alleles are capital R. Here you see, here small r. So only small r comes from here. Here capital R comes. When they come and fuse, they will be forming yes, capital R and small r. This is that first filial generation, or we say F1 generation. Now what will be that? What will be the color of this? That is what we are going to discuss now. Okay, well, let us see the first question here. Yes, first question says that state the color of the flower you would expect in the F1 generation. Now, F1 generation, what we got finally? From here, capital R and small r you got, right? This is the F1. Now, can you tell me what will be the color of this one? Capital R is there means what? What is the capital R for? Capital R is for red color. Alright. So, this capital R will be dominating the small r. That means small r, there is a allele. You can see there is a allele for white flower color, small r, but still the white r cannot express because capital R will be dominating. Capital R will be dominating the small r. So the capital R or the dominant character only will be expressing. That is what all plants will be with flower, red flower color. All plants will be with red flower color. So answer will be here. It is red. What will be the color of it? The color of the flower will be red. The color of the flower will be red here. Okay. Now, let us see the second question. Second question is that what must be the percentage of white flower, okay, in the F2 generation? Now, simple question. How do you get the F1? I mean, we got the F1 now. How do you get the F2 generation? It simply means this is our F1, right? F1 cross with the F1. That is what, when you cross the F1 with F1, that is what we call selfing. Okay, now let us go for selfing because then only you will be getting the percentage of white flower. Okay, let us go for the selfing. Now, this is what our F1 generation cross with the same F1, capital R, small r, cross with a capital R, small r. Okay, that is what is called selfing. Now, how do you get the gamete here? Here also you will be getting one capital R and here one small r. Here capital R, here one small r. Now, we need to make a Punnett square, right? So, let us make the Punnett square. Suppose this is the Punnett square. Here, in this case, okay, this gamete, these two gamete, capital R, okay, we arrange the capital R here, this is the small r, and these two gamete comes here, that is capital R and small r. Now what do you get? We need to mix the allele, right? Okay. Now here you will be getting capital R, capital R. Here you will be getting capital R, small r. Here you will be getting capital R, small r. Here you will be getting small r, small r. Okay. So now you will be knowing that capital R is there in the sense this will be of red color. Right. Here also there is a capital R. This will be red color. Here also there is a capital R. This will also be red color. Now, still you will be thinking, why there is a allele for white, but still it is red in color. That is what I said you. This dominant allele capital R dominate the small r. So, still they will be red color. Now, question is that what must be the percentage of white flower? Now, can you tell me here both the alleles for white flower? So, this is the only one flower which we can see with white. So, out of the 4, we have only white. Out of the 4, we have only white. That means what will be the percentage out of the 4? 1 is one white flower mean the percentage will be 25% of white flower. In the F2 generation, we will be getting only 25% of that. Clear? So that answer is clear. Let us go for the next question. Okay. State the expected ratio of the genotype of capital R and capital R small r. Okay. What we are going to get in the F2? In the F2 generation, what is the ratio between capital R, small r? Now, that is what we made here. After the crossing, finally we got like the Punnett square. It was like this: capital R, small r, capital R, small r. Here we get capital R, here we get capital R, small r, here we get capital R, small r, here we get small r, small r. Alright, now what is the question? What is the expected ratio of capital R? Now you just count how many capital R is there out of the 4. Only one. Only one capital R, right? Okay. Is to how many capital R, small r you have? Yes, this is 1 and this is 1. You have 2 capital R, small r. So what will be the ratio here in the F2 generation? In the F2 generation, the ratio between capital R 1:2 and capital R, small r 2. This is the ratio we are going to get in the F2 generation. Hope it's clear, my dear students. Okay. Let us go to the next question. Yes. Here, look at the question. Here it is given, name the plant on which the Mendel performed his experiment. Because we have studied that there is a particular plant on which the Mendel is performed his experiment, right? What is the name of the plant? That is what we have discussed, garden pea. What is that? This is the common name of that, garden pea, and this is the scientific name of that plant, Pisum sativum. Pisum sativum, this is the scientific name of that, and this is the common name of that. You can see the plant. This is the plant. This is the plant the Mendel used, garden pea or Pisum sativum. Okay, let us go to the next question. Yes, what is the next question? Next question is that what are the components of DNA? Just now we have seen the DNA, right? DNA is the double helical structure, right? Just now we said that. Look at the DNA structure now. Yes, this is the DNA structure. Now in this you can see this is the one strand and this is the another strand. Alright, now question is that what the DNA composed of or content? What is the content of the DNA? Now if you take the structure of the, yes, you can see the detailed structure of DNA. Look at here, in this DNA, this is the one strand and this is the other strand of it. As I said you, this is one strand and this one will be another strand, right? Now it has 2 strands. So in each strand you can see it's made up of, yes, this is what we are going to see. DNA, it's made up of many nucleotide. What is it called as? Nucleotide. So many nucleotide means, that's why it is given polynucleotide. What is meant by poly? Poly means many. Many nucleotide. Now can you see what is nucleotide then? So you will be having a confusion. What is nucleotide? This is one nucleotide, you can see. This is the one nucleotide. Means one nucleotide mean what all things are there? One nucleotide, it composed of nitrogen base. Please note it down. This is the nitrogen base. This is the nitrogen base. This is the nitrogen base. Now, nitrogen base mean who all are there? Sometime nitrogen base can be A means adenine. Sometime it can be A. Sometime it can be A. Sometime base can be T means thymine. Sometime the base can be C means cytosine. Sometime the base can be G, and that is guanine. So the base can change. This base, it can change. It can be A, it can be T, it can be C, it can be G, any one. Okay. So here suppose now base is A, adenine is the base. Okay. Now what you have in the polynucleotide? You should have a sugar. This is the sugar. You can see this is the sugar. This is the sugar, and there is one phosphorus also. One phosphorus. Where is the phosphorus here? This is the phosphorus. So, you should understand one sugar, one phosphorus, one base, together we call one nucleotide. What we call? One nucleotide. Now look at here. Look at here in this. This is one nucleotide. Right. This is another nucleotide. This is another nucleotide. That means each DNA consists of many nucleotides. That's why it is clearly said poly. Do you remember? It said that polynucleotide. This is the one nucleotide, right? So polynucleotide, like this many nucleotides are there. You can see one nucleotide, the other one. Right. So like this number of nucleotides arranged one after the other. That is what the chemical component, or we say components of DNA. That is the components of DNA. Let us go to the next question. Yes, what is the next question says? Describe the different type of chromatid, different types of chromosome. Because what is chromosome, as we have discussed, which is present inside the nucleus, right? So let us see what are the different type of chromosome. Yes, first one we are going to see, this is the chromosome. What we are going to see? Yes, first one, it's called as metacentric chromosome. What is metacentric chromosome? Here, a chromosome with centromere near the middle. Look at here. Now, this is what we call centromere. What is that? This center one. This is that centromere we say. Now, here you look at that centromere. Centromere is almost at the middle. Alright. Now, two chromatids are equal length. Now, what is chromatid? Chromatid means these two. These are the chromatid. Can you see? These are the chromatid. These arms of the chromosome, what we call chromatid. Now, in this type of chromosome, I mean in the metacentric chromosome, centromere will be at the middle, and those two chromatids will be almost equal in length. That type of chromosome we call metacentric chromosome. What is it called as? Metacentric chromosome. Now, let us see the second type of chromosome. That is the submetacentric. What is submetacentric mean? Here, centromere is situated slightly closer to one end. Okay, not exactly at the middle. Not exactly at the middle. You can see it is slightly closer to one end. It is not the middle, right? It is not even at the end also. That means it is slightly closer to the one end. So because of that, what will happen? One chromatid will be slightly longer. Look at here. This chromatid will be slightly longer, and this one will be little lesser than that. That is what is submetacentric. Now, what is the third chromosome? What we are going to discuss, that is acrocentric. What is acrocentric chromosome? In the acrocentric chromosome, centromere is situated near one end. Look at here. This is that. Near one end of that. And thus, you can see one chromosome is very long. Look at here. This chromatid will be very long, and this chromatid, you can see, it is very small. Very small. That type of chromosomes are called as acrocentric chromosome. That is the acrocentric chromosome. What is the last type of chromosome? That is telocentric. What is telocentric? Here, centromere is situated at the tip of the chromosome. Look at here. At the tip of the chromosome, centromere is there. So you can just see, the centromere will be at the tip of the chromosome. That is what is called as telocentric. So there are 4 different type of chromosome: metacentric, submetacentric, acrocentric, and telocentric chromosome. Okay, let us go to the next question. Yes, next question. What is monohybrid cross? Now you know it. Mono means what? What is the meaning of mono? Mono means single. Single what? That is what we are going to discuss here. Yes, it's a cross between one pair of contrasting character. One pair means what? 2. One pair means 2 characters. That means crossing 2 characters. One pair of contrasting character. Let us take a simple example, because it's given that give 2 examples. What are the examples? Yes. When you cross, suppose if you cross a tall plant with a dwarf plant. You can see here in this case, you see, we are crossing a tall plant with a dwarf plant. One is tall, one is dwarf. That means 2 characters. One pair of character we have taken and crossing, then those cross, what we call, it's called as monohybrid cross. What is it called as? Monohybrid. Let us see another example. Yes, we have taken another example like a plant with round seed with a wrinkled seed. You know what is that? We have taken here shape of the seed. Not the height. Before that, we have taken the height of the plant, right? Here we have taken the shape of the seed. We have taken shape. Here you see a round seed crossing with a wrinkled seed. So like this, one pair of contrasting character if you cross, that is what is called as monohybrid cross. That is what is called as monohybrid. That is the monohybrid cross. Yes, that is the monohybrid cross. Okay, let us discuss the next question. Yes. Why did Mendel choose pea plant for his experiment? Because we said that, before that we said the name of the experimental organism, that is Pisum sativum, right? Pea plant. Now, why? There was many plants, right? Why Mendel has selected only this plant? There will be many reasons. Yes. First reason is that, these plants grow very quickly. Within a short period of time, their life cycle is very short. Within a short period of time, it reproduce fast and very easy to study. Very easy to study this plant. That is what, one of the reason he has selected that one. And what is the second reason? Yes, second reason you see why Mendel is selected this one. And that is yes, there are 3 reasons. First reason we have discussed. Second reason is that it can be easily self-pollinated or cross-pollinated. Look at here. Now here, as I said you, in this case, this plant, it can easily self-pollinate. Between those, in the flowers of pea plant, it can be easily self-pollinated and it can be easily cross-pollinated. Cross-pollination as well as self-pollination, it was very easy to do. It was very easy to do. Either it is self-pollination or cross-pollination. It was very easy to do. Now, what is the third reason? Third reason, this garden pea has number of easily detectable contrasting character. Like how much? Almost 7 pairs of contrasting character. 7 pairs. This pea plant had 7 pairs of contrasting character. Look at here. First pair. Here, if you take the seed shape, the round seed is also there and even wrinkled seed. If you take the, color of the seed, there was some which was giving yellow seed color. Some of the plants were giving green seed color. When you take the pod shape, some of the plants were inflated pods and some of them were, producing constricted one. And when you take the pod color, some of those pods were green in color, some of them were yellow color. Like this, many different, that means 7 different pairs of characters were there. That is the reason, Mendel had selected this plant. So first one, they grow quickly and easy to study. Second reason was, it can be easily self-pollinated and cross-pollinated. Third reason was, almost number in the sense, 7 pairs of contrasting characters were available on the pea plant. Okay. Let us go to the next question. Yes. Here, see, Mendel crossed a tall plant, okay, with a dwarf plant, okay. Now, no medium-heighted plants were obtained in the F1. No, look at here. Here, Mendel, what he has done, he has crossed, you can see, he crossed a tall plant with a dwarf plant. And we have seen in the first example, after crossing these two, our Mendel got, capital T and small t. This was the F1. And we said all plants were tall. Because you can see here capital T, that means the dominant allele will dominate the recessive allele. That means small t cannot express. All plants were tall. All these plants what he got, that was tall. And question is that why he did not get any medium heighted plant? Normally what we think, a tall plant is crossing with a dwarf plant, we may get medium heighted plant. No, he did not get any medium heighted plant. What was the main reason? Yes, here reason was this one, dominant allele. Now, what is that? F1, this was the F1, capital T and small t, right? Now, here dominant allele mean our capital T, right? Capital T express themselves and prevent the recessive allele. Recessive allele mean our small t is recessive allele and capital T is the dominant allele. Now, here dominant allele were completely dominating over the recessive allele. They, capital T did not allow the small t to express, did not allow fully masked, fully hidden the small t. That is what here in the F1 generation, only dominant allele is expressed and the recessive allele is suppressed. So you will be thinking like, then why this small t and small t, why they are not expressing? Small t when two recessive allele will express only when they come in a homozygous condition. If they comes like this, small t condition, only that time they will express. That time it will be of short. Otherwise, even one capital T is there, still the plant will be tall. Are you getting the point? So here reason is that dominant allele is completely suppressing over the small recessive allele. That is the reason he did not get any medium heighted plant. All plants were tall. Okay. Yes, you can see here in the F1 all the plants were tall. There is no medium heighted plant. Okay, let us go to the next question. Yes, here. Father is responsible for the sex of the child and not the mother. Justify the statement. Normally, father is responsible for the sex of the child. So now let us prove it. Actually, normally the people talk about mother is responsible for the sex of the child. No. Why it's not? The mother, the chromosome will be, X and X. In the female it is X and X. We have already discussed in the sex chromosome. And in the male, the chromosome is going to be X and Y. Okay. Now, what is the role here? Yes, look at here. In this case, this is the male. Okay. Male has the chromosome XY. Now, when the male produce the male gamete, when they produce the sperms, half of the sperm or some of the sperms will be having X chromosome, some of the sperms will be having Y chromosome. Right, but look at the female, their chromosome is XX. That means they will be producing only one gamete, and that also once in a month. Egg will be producing once in a month, one egg will be producing, and that chromosome is with X only because they have both the X chromosome, right? Both the X chromosome. So here you can see female will be producing the egg with X chromosome. Now who determine who is going to fertilize? Now suppose this sperm with X chromosome is going to fertilize. Fertilize means fusion. Who is going to fuse it? Now, if this sperm with X chromosome is fertilizing, then surely X from this sperm, X from the egg, they form a XX. That will be, XX means what? XX will be female. So, they will be producing a female. Or in this case, the sperm with Y chromosome is fertilizing with the X chromosome egg, then surely X and Y will be producing and the offspring will be male. So who is responsible here? In which, which sperm is going to fertilize? That is the matter. Which sperm is going to fertilize? That is the matter. And the mother does not have any role. Anyhow, the chromosome of egg will be X. No role in that. Female does not have any role. Here, who determine which sperm is going to fertilize the egg? So that is what it is clearly given here. Father is responsible for the sex of the child, not the mother. Not the mother. Okay. Let us go to the next question. Yes. What is the next question? Next question is that explain dihybrid cross. Now, what is mean by di? Di mean two. Before that, we have discussed monohybrid cross, right? One pair of contrasting character. Here, dihybrid cross in the sense two pair of contrasting character, and we have to, you have to give a example. Okay, let us discuss that. First, what is dihybrid cross? Yes, it is a cross between two pair of contrasting characters. Two pair means what? Two pair means four. Four characters we have to cross. Okay. Now let us see here. We are crossing there. We are crossing round yellow. Okay, a round yellow seed. That means the shape of the seed is round and color of the seed is yellow. Now it is crossing with another seed which is green in color and wrinkled. You can see the shape of that. That is a round yellow seed crossing with a green wrinkled. And whenever we cross it, we know it, whenever we cross, we are going to get the F1. What is the F1? Here you will be getting capital R is for round and small r, and capital Y and small y. This will be the F1. This will be the F1. That means capital R is for what? Round. Okay. And capital Y is for what? Yellow. And because of the presence of, you can see here capital R is dominant, so surely it will be red, and capital Y is there, so surely it will be yellow. So what will be the phenotype of this one? You can say the phenotype of our first F1 generation will be capital R, small r, capital Y, small y. This will be the F1, and this is going to be round yellow. All plants in the F1, it will be round yellow. Now, what we are going to do now? We need to do the selfing, right? Selfing means what? Our F1 into F1, capital R, small r, into capital Y, small y. This is what is called as selfing. Now, when you do the selfing, look at here. When you do the selfing, yes, we are going to get like this appearance. You can see here when you take, because when we are getting the gametes like capital R, small r, capital Y, small y, into the same capital R, small r, capital Y, small y. Okay. Now, how do you get this gamete? Look at here. Here there are many gametes. Now, how do you get these gametes? These gametes we are getting, I'll tell you easy way to produce the gametes, like first capital R goes to capital Y. Okay. Second case, capital R goes to small y. Capital R goes to small y. In the third case, what happened? The small r goes to capital Y. Small r goes to capital Y, and small r goes to small y. Small r goes to small y. So, this is also the same parent, right? So, that means even they will be getting the same gamete. Capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Even they will be having the same gamete. So, one side we will be getting 4 gamete. Even this side we will be getting 4 gamete. Now, what we need to do? We need to make the Punnett square, and this is the Punnett square what we made. You can see. Yes, 4 gametes here and the 4 gametes here. After crossing those 4 gametes, you will be getting, these are the new offspring. We are going to get, there are 16 new plants. Out of that 16, plants, we are going to get, you can see, you can just count. This is a round yellow. This is round yellow. Even, you can see this is a round yellow. Even this is round yellow. So, the number of round yellow seeds will be, you can see 9 round yellow. And you can see there are 3 round green. 3 round green. So, here ratio will be 9:3:, same way three, you can see there are three yellow wrinkled. There are three. So you can see there are three yellow wrinkled, and last one which is of green wrinkled. So this is the phenotypic ratio of dihybrid cross. According to the phenotype, we got 9 yellow round and 3 green round and 3 yellow wrinkled and 1 green wrinkled. This is what is called as phenotypic ratio of dihybrid cross. Okay students, hope you got all the questions what we have discussed here. We'll meet in the next class with a new topic.
Brilliant Qatar, your trusted coaching partner for IIT, JEE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.